My name is Ryan Simkowski, and I am a project scientist at the UC San Diego. And today we're going to talk about metabolic engineering of cyanobacteria, ultimately for the purpose of making production strains that are capable of producing from sunlight to high-value products such as oils, lipids, proteins, pharmaceuticals, etc. Before we get too far into those weeds, however, let me first remind you what cyanobacteria are and what we can do with them. So cyanobacteria are a diverse group of oxidative photosynthetic prokaryotes. That means that they're able to convert sunlight and carbon dioxide into carbon-based organic chemicals. We have a number of genetic tools that have been developed over 35 years in order to be able to alter protein expression levels, functions, single base changes, etc., to produce things such as knockouts of proteins or overexpressions of proteins, or simply changes in functionality. And you can see an example of that in the figure at the top, where Synechococcus elongatus, a unicellular cyanobacteria, has been altered to express YFP so that it can express in a yellow or green channel of a fluorescence microscope. You can also see another example wherein we can choose specific types of regulatory elements in order to have specific types of cells or specific conditions enable protein expression. So at the bottom you can see a filament of Anabina, a filamentous species, where specifically the heterocyst cells are expressing YFP. Those are cells that have differentiated to be able to do nitrogen fixing. And here only those cells are expressing the YFP due to the reporter construct. Now, a number of cyanobacteria have been engineered to generate biochemical products that are of industrial value. And here's just an example of those kinds of products that are highlighted with the colors. So things like isobutyraldehyde, ethanol, exportation of sugars and other high-value commodities that can eventually be able to be used to replace petroleum products or petroleum-based products. But there's a problem with this. Although we've been able to use the genetic engineering tools that we've discussed in a previous lecture to accomplish this, often the yields are pretty low. And there's a number of reasons that this could be due to. First of all, you're trying to express a chemical that may not be compatible with that strain. But alternatively, there's issues such as expression levels of the proteins that enable those exogenous commodities. Here in this figure, those commodities are highlighted in blue, so isoprene, ethanol, etc. Additional genes have been added to be able to make those. Alternatively, there may be toxicity issues, or the other potential problem is that carbon simply isn't flowing towards that output, or the cell is responding to the fact that we're now causing the cell to make those products by shutting those pathways down. So the question is, how do we then increase yields? One method is something that we talked about in the previous lecture on genetic engineering, which is that we tinker with how much the enzyme is being made and try and express it as best as we can in an optimal fashion as we can. Alternatively, that's where we're turning to metabolic engineering. How can we alter these pathways to direct the flow of carbon towards our end product? So, how do we go about doing that? First, we have to know all of those pathways. So we need to have a large body of knowledge about cyanobacteria in general and their metabolic pathways. Now, even in the most well-studied systems, such as E. coli, about 30% of the genes still have unknown functions, so there is still gaps in our knowledge base. However, we do have quite a number of tools now to be able to acquire this kind of information fairly rapidly and exhaustively. So omics studies in systems biology, such as genomics, transcriptomics, proteomics, and metabolomics, really help us understand both our model systems and new novel systems extracted from the environment to be able to better get a metabolic map and a model to be able to predict where it is that carbon is going to flow. So as an example, in this figure, you can see that some carbon is going to be flowing towards glycogen at the top, whereas other carbon is going to be flowing down towards the TCA cycle to be able to make succinate, which was actually the product that this particular group wanted to enhance. So, based upon these models, both the map as well as mathematical information about the flux through these pathways, we can predict where it is that is best to try and make stoppages in the flow or redirections in the flow. So you can see at top, GLIG-C is either downregulated or deleted, and those kinds of deletions will then prevent flow of carbon towards an off-pathway carbon sink that we don't want it to go towards and is causing yields of our products to be low. So that's one strategy. Other strategies are to enhance the expression of those pathways that go towards the product we want, or again, prevent flow away from the product itself. So for instance, knocking down SDHA in this figure at the bottom left would prevent the flow of carbon around the circle, and they would start to accumulate at succinate. So these are successful strategies that people have employed, but even still, these strategies don't always give us the optimal yields that we need in order to be both industrially relevant and economically feasible. And so again, the success of the strategy is knowing more about the metabolic system. Because again, we often find there are surprises. There are new pathways that we didn't know about that get activated when we start tinkering too much. So there are now novel tools to be able to try and get a better understanding and a more complete understanding of our cyanobacterial systems. So out of the Golden Lab here at UCSD, a new tool has been developed from systems developed up in Berkeley, wherein we are generating a barcoded transposon insertion library. What that means is that, as demonstrated in the upper panel, we take a transposon library where each transposon is uniquely barcoded, and we transform or conjugate that into Synechococcus elongatus. That then marks individual cells with one single transposon because of the way in which the experiment is performed, so that each cell has its own unique barcode associated with it, and that barcode goes into a unique position in the genome. We can then sequence where all those barcodes are located within a single pooled population, and then map where those insertions are located and their associated barcodes back to the genome. And that's what you're seeing on the bottom. The green lines around that circle represents all of the insertions we are able to map back to the genome of Synechococcus elongatus, specifically the chromosome. We also have insertions in its plasmids. And the red bars are where genes are located. And this particular library was very well covered. So we have over 250,000 insertions for a 2.7 megabase genome, which means we have on average one insertion every 11 base pairs. And you can see with the blue arrows on the upper part of the lower panel, where it says SYNPCC 7942-1241, that this is just a region of genes where these genes are not essential, and you can see the places where insertions are located, and they're pretty uniformly distributed all throughout those genes. Again, about once every 11 base pairs. In other regions, there are stretches that are much larger than 11 base pairs that lack any insertions, and you can see that in the bottom part of that panel where there's the red colored genes, such as RPOB. RPOB is an RNA polymerase, and it is essential for the organism to be able to function, for the organism to live. So any insertions that went into that gene cause that cell to die. And so we don't see it when we end up growing the pool of the library and end up sequencing it. So these large gaps can tell us where there are essential genes or essential regions of other functions, so non-coding RNAs or small RNAs, things that we simply haven't discovered previously. So this information allowed us to then determine what the essential gene set was for Synechococcus elongatus, which is really important information for metabolic engineering. You don't want to waste your time trying to knock out a gene that simply can't be knocked out, that the cell needs in order to be able to grow and divide. So as it turns out, a little over a quarter of the genes in the genome are essential. The rest of the genes either have some mild effect if they're mutated or are simply non-essential. We can get rid of them completely and it does not seem to harm the cell under laboratory conditions. We can then take that information and map it back to our metabolic maps. And that's what you see on the right figure. The red boxes represent essential genes going around the central carbon circle. And then in the blue boxes are those that are non-essential. And so, of course, here, again, if we wanted to optimize succinate production, we would know that we don't want to do too much to remove the production of those red boxes, but we could easily remove the blue boxes in order to enhance succinate production again. So this is very important information for us to know, but even more so, this TNSeq tool has another power, and that power is phenotypic evaluation. So what we can do is take the pooled library. We can once again sequence it just to get a baseline, and that's what you see on the far left of the first figure. Then we can split that library into multiple different growth conditions. In this case, the growth conditions are in photobioreactors, either under continuous light, LL, or light-dark cycles, LD. Now, in the lab, we most often grow our algae and our cyanobacteria under continuous light, simply because it makes them grow faster, grow more dense, and we can do more stuff with them, more experiments. But in a real environment, if we're going to be growing these outdoors, we need to make sure that they're able to survive light-dark cycles, because that is what's going to happen in real life. So this particular experiment was performed with the library to understand which genes were very important for being able to survive light-dark cycles. And you can see a figured result in the bar graph off to the right. So the blue gene would be one that, when mutated, actually enhances fitness in light-dark cycles. So its native state is inhibiting growth under light-dark cycles, whereas the orange bar is one that goes way down in light-dark cycles, and that would be one that would be considered essential for light-dark survival. I'll show you more results about that experiment on the next slide. Another thing that we can do with this particular tool, it's pretty much one of our hammers that we can now use to try and understand as much as possible about our cyanobacteria, is to introduce a second mutation. So in this particular case, we introduced a second mutation in panel B, either a control mutation or experimental mutation of a gene that we're interested in. And now we can start to see synthetic lethalities. So when it is that the mutation that is occurring due to the transposon, the barcoded transposon insertion, is interacting with the mutation we now introduce in order to cause lethality. That's the first experimental knowledge that we get from this particular experiment, but then subsequent to that, we can once again take the pooled library, split it into different environmental conditions, and once again ask, under the condition of having multiple mutations, are we getting synthetic lethality under the experimental condition, in this case light-dark, for example, that we don't otherwise get under continuous light experiments. So this gives us quite a lot of information about how the metabolism is functioning. And it's, again, very important information for metabolic engineering, where most often you're creating multiple mutations in metabolic pathways, and you want to know how they are interacting with each other. This tool is going to be a great tool for being able to understand that a little bit better. Okay, so I said I'd go through some of these results with light and light-dark because it is of particular relevance to being able to use these production strains out in an environmental scenario, especially where we're using light as our source for energy and carbon dioxide as a source for carbon. So in the first panel, you can see that out of all of the genes that were investigated, only about 220 of them ended up being important for survival or for doing better under light-dark growth conditions as compared to light. And that's the Venn diagram at the top with the colored bars. You can see that there's a number that have only moderate effects and some that have strong effects, either positive or negative. That data can also be shown on the bottom panel in B, where we're taking the fitness score that we calculate for each gene. So how much does that mutation impact each gene? Or actually, it's a set of mutations impacts each gene. How does that impact it versus a statistical significance value, the FDR, here, false discovery rate. And so we know how significant these truly are based upon the statistics of doing multiple experiments, and are able to determine what's real, what is not real, what's a true impact, for example, essentiality under light-dark, and what is not. And so based upon all of that data, we can then identify which genes are necessary and which ones, in fact, aid growth under those conditions. We can determine what kinds of functionalities we know about, and otherwise we can map these back to the metabolic maps that we've discussed before, which is what's being shown on the right. So again, on the right, you see that any arrow that's colored in red is essential in light-dark, but was not essential in light-light. Those that are dashed were essential in light-light, so they were once again essential in light-dark, since everything started under light-light conditions. And then black are those genes that are non-essential. So once again, it tells us what can we change under a light-dark cycle, for example, now, and that's information that we did not know before when we were only doing the experiments in continuous light. So again, this is really important information for us to be able to do the metabolic engineering that we want to do. And that kind of information can be incorporated into other strategies that people have used in order to optimize product yield. So for example, a completely different strategy is taking cyanobacteria and being able to grow them heterotrophically. So for the most part, most people are thinking, well, cyanobacteria are photosynthetic organisms. Most are naturally obligate autophototrophs, meaning they can only grow from sunlight. They cannot take up sugar or carbon, other carbon sources in order to grow. And that means at nighttime, they're going to be essentially quiet. They're not going to be able to produce our product. So the Atsumi group at UC Davis came up with the idea of how about we engineer them to be able to take up carbon so that during the dark parts of the day, they can use carbon as their feedstock for their metabolism. And then during the light part of the day, they can use carbon dioxide and sunlight in order to keep making the products that they want to make. So they engineered into Synechococcus longatus a number of different sugar channels, each individually into an individual strain, either for glycerol, xylose, or glucose, as you can see in the figure on the bottom. And this allowed for the cyanobacteria to grow heterotrophically in the dark and use those sugars to be able to continue to do chemical production. And what they found is that it was, in fact, able to increase production under light-dark cycle conditions to be equal to that that they get under continuous light conditions with a non-engineered strain. So basically, they were able to keep the metabolism running all through the day, even though the lights turn off. Then they were able to make mutations that would otherwise not be allowed because, for example, in the glycogen storage pathway, because the cell would have needed that without any other sugar energy source at night. So now they can make extra mutations that then allowed them to increase their yields about fourfold to about 12.6 grams per liter or 53 milligrams per liter per hour, which, although is still not quite economically viable, is at least starting to get into to the arena of being able to be marketable. It gets us one step closer, that's for sure. So I hope you've learned quite a bit about how we do metabolic engineering and what kind of tools we can use to be able to understand the cell a little bit better. If you are interested in learning more, I've listed a number of papers here that are quite interesting and can provide a lot of information about the tools that I've talked about, as well as metabolic engineering and cyanobacteria in general. Thank you for listening to the course, and I hope you listen to some more of the videos.